Good afternoon. You're here on Likeable Science. I'm your host, Ethan Allen. We're broadcasting from the ThinkTech Hawaii studios in Pioneer Plaza. And Likeable Science is all about helping people understand and appreciate that science is a fun, vital, and interesting part of their daily lives. It's not something to be intimidated by, to be avoided, to be run away from, but it's very neat stuff. And I thank you for joining us. Today, to help talk with me about science is Joshua Copus. And Joshua is a graduate student in Hawaii's Institute of Marine Biology at the University of Hawaii here. Welcome, Josh.
Thanks for having me.
So, Josh does some interesting work, and we're calling this show Diving into Research because Josh's work actually involves him getting out, going to 400, 450 feet sometimes in the pursuit of his particular research, which we'll talk about in a bit. So, but I thought we should start, Josh, by sort of clarifying a few terms. In our earlier exchanges about your work, you used some terms like biogeography and phylogeography. Can you maybe parse those out a little bit for us?
Absolutely. The difference between biogeography and phylogeography is biogeography really is how species are distributed geographically. Okay. Phylogeography looks at the relationship of those distributions, so how species are distributed and how one population is related to another. Okay. So that's essentially the difference between the two. They both focus on the geographic distribution of species.
Right. So how many fish of what kinds are where and sort of why, more or less.
Exactly, yes.
Oh, okay, well good, that's great. And you in particular are interested not in the fish that swim around at the surface, right? You're interested in these fish that swim in the so-called mesophotic mid-light region, right?
Yes and no.
Okay.
I'm interested in the biogeography of all coral reef fish. Okay. But my research really is largely focusing on the mesophotic fish because there's nothing really known about them. And they may actually have a different evolutionary history than the shallow fish. We don't know. Mm-hmm. There are some patterns that we're starting to see as we dive deep that may suggest that they have different biogeographic patterns than the shallow fish. But it's still a very, very new area of exploration, so we're not quite sure actually what we're going to find. And so I do a lot of mesophotic fish work for that reason.
I see. So yeah, we don't often appreciate, I think, how much of the world is sort of reasonably deep ocean. I saw a little sort of infographic the other day that points out that our image of the ocean is either this blue surface or some nice pretty corals under it, seagrass beds, whatever, really represents only a tiny fraction, and indeed 94, 96% of the ocean is basically black, utterly black abyssal depths.
Absolutely, and actually, if you're talking about coral reefs, the idea that you get in your head is that, they're shallow.
Right.
But really, coral reefs in some places in the Pacific can go down to 600 feet or deeper. And so, because of the limitations of our scuba diving and such, it's really only the 90 to 100 foot and shallower that have been studied. So a good 3/4 of coral reef habitat is largely unexplored.
So then you really are actually out in cutting edge territory here.
Absolutely, absolutely.
That's very exciting. And again, it's the issue on these deeper reefs, or at least one issue is, of course, you're losing light as you go deeper and deeper steadily. And you were saying earlier that sometimes, depending upon the time of day, the lighting conditions, the water, you can get reasonable amounts of light there, although I think you've probably lost a lot of color.
Yes.
But other times it's quite dark.
Yeah, and it is really very specific to each location that we dive. But in some places you can be, 100 meters down and you can still see reasonably well. And you're right, color does get filtered out as you go down deeper. But other places, if you don't have a flashlight, you're pretty much in trouble. It's dark.
Yeah.
You don't get to see too much while you're down there.
Right, and yet there are thriving communities of fish. You were saying earlier that you you go out, I guess, for a couple of weeks at a time on some vessel, and you say that during these dives you routinely discover new species of fish.
Yeah, and actually most of our expeditions are shore-based. We don't actually go on vessels. We'll rent small boats and take them out on the reef, but we typically go to remote locations that haven't really been studied all that much. And so we usually get in about 14 or 15 dive days, and in that time frame, we'll come up with at least a half a dozen potential new species of fish, sometimes more.
Yeah, that's stunning to think that there are so many undiscovered species of fish. We tend to think, well, we pretty much know all the vertebrate life forms on the planet here, but again, it's telling of our lack of information about the ocean depths that you can do this.
Absolutely, and actually the mesophotic depths may have more undiscovered species than even deeper depths, and the reason for that may be that, you know, if you're going to dive that deep, traditionally you would have to use a submarine or ROV. But subs are very expensive to operate, and if you're going to operate one, you're not going to just stay in that shallow 100 to 150 meter depth. You're going to go really deep.
Right.
And so we have a decent amount of information below the mesophotic, but there's this gap in our understanding, and it's largely due to technological limitations.
Right, and the real abyssal depths may be something, at least in some areas, something of a desert, basically, right?
Yes.
They may have a relatively sparse life forms and all, as compared to mesophotic, which probably
Right.
yes, is richly endowed with species.
And the deeper depths all have been largely surveyed with different nets and things like that can capture those fish. But in the mesophotic depth, you have a lot of caves and undercuts and stuff where really the cryptic small species that we're finding, they're all really small, nothing that you can catch on a hook and line or large fish, but mostly what we're finding are these small cryptic species. There's really no collection method that you can use from a boat, a net or so forth to collect these. So you actually have to go down there in person and capture them.
So how do you do that?
Well, so our team uses what's called a closed circuit rebreather. And so everybody's heard of scuba diving, and the difference between scuba and closed circuit rebreather is scuba, you inhale the gas in your tank and you exhale, and all of those bubbles go out into the water. You lose a lot of the volume of gas in the tank because you don't use all the oxygen that you've inhaled, so you're exhaling some of the oxygen. So the idea of a rebreather is that it captures the exhaled gas and it sends it through a CO2 scrubber to remove the CO2, which can kill you, and then you can rebreathe it.
Right.
And it has a computer that monitors the oxygen levels, and when you breathe the oxygen too low, which can also kill you, it adds more. And so it kind of maintains this really ideal mixture of oxygen. It removes the CO2 and gives you the maximum amount of gas. So you don't have to carry as many tanks.
Right, and so you can stay down for longer periods of time, because just to get down there must take more than a few moments, and then going back up, you have
Getting down is relatively quick.
But
It takes as long as it takes to swim there.
Okay.
You don't have to go slow as you descend, but coming back up, it takes quite a bit of time because you have to worry about the bends,
Right.
which a lot of us have heard of. And so we have to, a lot of our dives are 4 to 5 hours of decompression time before we're allowed to come back to the surface.
Wow.
And that's just to allow for the inert gases in our body to come out slowly instead of in bubbles, which will, again, kill you.
Right.
So.
Well, and that's why I've got to use a rebreathing system, because you have to make reasonably efficient use of the
Right.
gas. You can't carry 4 to 5 hours worth of
So yeah, there are
That's blowing
people
away.
that use open circuit scuba to do these kinds of dives, but they do have to take a lot of tanks.
Right.
I think there was a record, new deepest diving record that has just come out a couple weeks ago, and I saw a picture of it. I didn't really read the article, but he had 4 or 6 tanks just strapped to his back, and he must have had quite a few staged for his trip back up. I mean, it's an incredible amount of
Right.
gas that you would need for open circuit.
Wow. But so you're then down sort of on the face of a coral reef with nooks and crannies and caves, and you're trying to catch small fish. So, I mean, how do you do this? You can't just sort of whistle up and say, hey, come here, little fish.
So we use really two methods to capture fish. Hand nets, so we
Okay.
chase them around with nets, and some of us are better at that than others. We also, if the fish is large enough, I mean, we can get fish in this size range as well. We can use a hand spear.
Huh.
And it really depends on what we're going to use the fish for in the long run. Do we need to collect it with a net, or can we use a spear?
Okay.
So those are our two methods. So we typically chase fish around with nets or spears and attempt to collect them. And when you're diving that deep, you have a very, very limited bottom time, because every minute you stay compounds to, depending on your depth, quite a few minutes of decompression.
Right.
When we're down real deep, I mean, if you stay an extra minute or two, it could mean another half hour or an hour of decompression time.
Hmm.
So you're always in a hurry to get down there, to do your work, and then you have a long time to think about it on your way up,
Huh.
because you're just,
And there's not much to do on the way up.
you get to look at fish and look at then all the neat stuff that are on the coral reefs. But yeah, you spend a lot of time taking pictures, and there may be other fish that we're interested in collecting, and we'll collect them on the way up. We also do fish counts, because we're not only interested in what's down deep, but we're also interested in the shallow,
Right, how it transitions.
how it transitions, exactly.
So some of these fish, if you catch them in a net, you presume then put them in some sort of little mesh bag or something, and
We'll put them in a collection bucket, because
Okay.
we'll want to bring them back up alive.
Right.
And they have to decompress
Right, I was
as
gonna
well
say,
as we do, but it generally, if we just take them up on our decompression schedule, they probably wouldn't make it. So you have to actually needle their gas bladder. And so fish have a gas bladder that allows them to stay floating in the
Right.
water. And as we come up shallower, that gas expands, the same with the gas in our body. And unless you remove the gas from their gas bladder, they won't be able to stay upright, and then eventually they'll die. And so we actually take a little hypodermic needle, and we remove some of the air out of their gas bladder at different stages on the way up.
wow, okay. So it's a little more elaborate than I was envisioning it. Huh?
Yeah, it's quite a process to get work done down there, yeah.
Yeah, a very, very challenging environment, and one very unforgiving of mistakes too. As you say, if you have any good stories of exciting things that happen on your dives, you've done something wrong, right?
Either you've done something wrong, or so you rely very heavily on your equipment,
Right.
and equipment malfunctions.
Right.
And so it could just be that, you blew an O-ring, and
Right.
or there's a lot of different scenarios that can happen. And we have so much training that we're taught how to deal with the different scenarios, but there is still a certain amount of danger to the type of work that we do.
Right. Yeah. No, it's an odd sort of blend of technically demanding, scientifically demanding, very constrained for time,
Yeah.
quite with real dangers from the environment.
Yeah,
Very,
absolutely.
very intriguing.
But when you collect a new species of fish and you have it in your hands, and you're the first person that's ever seen that particular species, you know why you do it.
Yeah.
It absolutely
must be
becomes very clear.
Yeah. You're measurably sort of adding to human knowledge here in a
Absolutely, and
substantial way.
you're the first person
Right.
who's ever gotten to
Right.
see that particular organism.
That's great. Now, because some fish have quite elaborate life cycles and will go through all kinds of changes, switching from male to female or vice versa, do you have fish that you might initially not recognize that later turn out to be just a different, either a juvenile of a relatively more better known fish where the adults are known and people just haven't seen the juveniles, or that kind of stuff?
We don't typically run into those sorts of issues because, well, first of all, we don't know anything about the ecology of the fish down there. So if it's not found shallow, we really don't know. There are some issues of where we're not sure if that's a female and this other one's a male, And so the genetics will help us determine that, and morphology as well. But most of the time, if we think they're new, it's not because we think there's something that's shallow, but it's a different form. Because we'd also see that shallow. And the shallow reefs are fairly well studied.
Excellent. So we're going to take our first break here. We're on Likeable Science. My guest here is Joshua Copus from the Hawaii Institute for Marine Biology. We're talking about his interesting diving research into the mesophotic region of coral reefs and studies of the biogeography and phylogeography of coral reef fish. We'll be right back.
Aloha, I'm Kili'i Akina, president of the Grassroot Institute and host on Ehana Kako, a weekly program on the ThinkTech Hawaii broadcast network. Ehana Kako means let's work together. Think of the sad alternative, let's not work together. Here in Hawaii, with all of our diversity and the richness of the people, it's important for us to come together around issues on the basis of what's right and what's good and what's going to serve the common good. And that's what we try to do at Ehana Kako. Every week we interview movers and shakers, people in government, business, and other sectors of society to talk about how to create together a better government, economy, a better world here in Hawaii that can bless the rest of the world. I thank you for your attention to ThinkTech Hawaii, and we look forward to seeing you every Monday, 2 to 3 p.m. on the ThinkTech Hawaii Broadcast Network. We're Ehana Kako, and we wish you well. Aloha.
And you're back on Likeable Science. You're on ThinkTech Hawaii. I'm your host, Ethan Allen. With me in the studio today is Joshua Copus. He's a graduate student at the Hawaii Institute for Marine Biology at the University of Hawaii. We're talking about coral reefs, coral reef fish, and how he is investigating the various species, various families of these fish, where they are, why they're there, how some are there and some are other places, I guess, to put it in simple terms. But so we were talking in the earlier part about how you go out and collect these fish. So once you collected them, some of them you may be wanting to keep alive, you said, but others you just really want tissue samples from. So what's the next step then?
So once we've collected the fish, and we get back to the boat, then we take the fish back to the shore. And then we spend the rest of the evenings processing those fish. So we'll take photographs of them. We'll take the tissue samples, and we generally take genetic samples, but lots of times we'll take isotope samples or other types of samples as well. We try to get the maximum value for each individual that we collect. And we enter each fish into a database and give it a unique number so we can always trace that individual back to the tissue, back to the individual, and so forth. And then we preserve them for the museum. A lot of our stuff goes to the Bishop Museum. And that's a very lengthy process. And so we're often working till midnight or later at trying to get that day's work done so we can go back diving the next day.
Okay. And then at some point, though, you finish your collection run, and you're back sort of in your lab for a bit?
Yes, and so a lot of the work, so the diving and the collecting, that's a lot of the fun stuff to talk about, but a lot of the work actually happens in the lab. And I'm in a genetics lab, and we use genetic techniques to look at populations and species relationships. So we use genetics to look at how the new species are related to other species in the family, but we also we look at how populations are connected between locations, and so we do quite a bit of work in the lab to determine that.
Right. No, that's a big area, not just in fish these days. I mean, I was just reading a recent issue of Science. They've just completed a large study on birds where they've got multiple species from every major group of birds and did a complete genomic analyses on these and actually sort of restructured the tree of birds, as it were, in terms of what birds are related to how to which other birds. And so it's
absolutely,
yeah.
that's happening in every group of organism that's out there. with the genetic techniques, they've been around for a while now, but we still have a lot of catching up to do.
And there's a lot of information that can be gained. I mean, I was interested to see in the bird stuff that they can now trace back the origin of learned song, basically, in 3 different lineages of birds, basically, and it apparently really independently evolved 3 different times. And I'd assume in the fish world there must be similar sorts of things.
Oh, absolutely.
Yeah.
You can attempt to use genetics to trace back any
Right.
characters and behaviors to a certain degree as well.
Right. And yeah, so okay. So I guess where is this going? You know, where is this work taking you? What do you hope to get out of it?
Well, so my focus really is on Pacific and the biogeography of the Pacific. And there are a lot of hypotheses about why species are where. And my work really is to try to help tease out, you know, what is regulating species distributions across the Pacific. And, you know, what historical events may drive why certain species are where they are.
Mm-hmm.
There's some places that you would expect a particular species of fish to be because it's next door at the next archipelago, and it may be on one past it, but it's not found in a certain location. And so these different questions are, we still don't know. We don't know why exactly, what drives that biogeography. And so that's really the focus, the overall umbrella of my research.
So you look around Hawaii, but you also then look elsewhere in the Pacific?
Yeah, so I look within the Hawaiian chain.
Mm-hmm.
I have a project going on across from the main Hawaiian Islands all the way up to the northwest.
Mm-hmm.
But I also am very interested in the Pacific as a whole. And so we do a lot of, we just in June were in Ponape, Micronesia.
Okay.
And we were in the Philippines in December. And so we go to other places across the Pacific. And
we should talk more. My work, completely independent of that, brings me out to Yap and Chuuk and Palau and the Marshall Islands. So yeah, fascinating parts of the world. But
they're very neat.
Very remote little sets of islands, a long way from anything else. Yeah.
And very difficult to do research at,
Right.
and they're very understudied
Right.
compared to other locations like Hawaii, which has been largely studied, at least compared to places like Micronesia.
Right, and yes, as you say, lots of challenges there. If something breaks there,
You better
it's
have
broken.
a second.
Yeah, you better have
Yeah.
a spare if you need it,
That's right.
because you're not going to find it there, right?
Yeah, the challenges of conducting an expedition are such that I think we're the only group of people, of researchers, that are actually doing these remote expeditions across the Pacific. I'm not aware of another group that does what we do.
And so you talked about the factors that may cause species to be in one place and not in other places. And I'd imagine that can also change both over sort of geologic time, but it can also, is probably changing now because of the ways we are changing the ocean, basically, right? I mean, we are, one, adding a whole bunch of heat to the ocean and changing the environment even at considerable depths. And 2, we are raining microplastics through the ocean at a truly awesome rate.
Yes. Well, that's a very large and loaded question,
Right.
and we'd have to spend quite a few of these sessions to really answer it. But the short answer is yes. Humans do impact the species distributions by either removing a species from an area through destruction of habitat or overfishing, or by introducing species to a place that they currently weren't, through bilge water or through, just transplanting them.
Right. Is it the lionfish in the Caribbean now that have gotten well established and are wreaking havoc with a lot of the
Yeah,
other
that's
species
a
there?
very, very big issue.
Yeah.
And I believe was an aquarium introduction possibly,
Huh.
but I'm not sure
Yeah.
if they've confirmed that.
Yeah, that's-
But yeah, that one's a big issue. But there are also others. And you know, you mentioned the added pollution in the water, the added garbage and stuff. Larval fish, so most fish have a larval stage.
Right.
Where their eggs float in the water and then they develop in larvae and eventually they can settle out.
Right.
And so they spend a lot of this larval stage out in the ocean. But if they reach a point that they can settle, they may settle on flotsam or garbage that is floating in the ocean and then follow that into a place that they wouldn't normally have been able to get to because their larval duration wouldn't have been long enough. So it's possible that pollution can also cause species to get where they normally would not have.
Oh, okay. I hadn't thought about that. That's intriguing to realize it can hurt, but it can help in an odd way in some cases.
I don't know. It depends on who you would talk to, whether that's actually helping or not. Introducing species where they don't belong, a lot of people would say is not actually
helping in the sense helping that species
helping
expand.
that individual survive.
Right.
Correct.
Yeah.
That would definitely be a help.
Yeah. So that's most interesting. And so it sort of seems like there's massive sort of never-ending studies that could be done.
Yeah, and so right now I'm focusing really on how sea level change through glacial cycles can really drive species distributions. And so sea levels fluctuate on about 100,000 year cycle, and at about an amplitude of 120 meters or so. So there's a considerable change in sea level during glaciation events, when the water is trapped in glacial ice. And so the reason we believe that, or one of the reasons we believe that these mesophotic fish may have a different evolutionary history is because they're below that 100-meter sea level fluctuation, and they may not be subjected to the same pressures as the shallow water fish. And so they may have more stable populations and may be able to persist in remote locations for longer and able to speciate and so forth.
Excellent.
And so right now I'm really trying to focus a lot of my research around how the patterns we see and how sea level change can inform those patterns.
Right, because now sea level rising, as it is, it's sort of the other extent is happening. Fish at the surface are able to colonize areas that they couldn't colonize before. You know?
Exactly, fish and other organisms as well. And so as the sea level rises, you may gain more habitat in a particular area, which may increase fish populations, or it may open up different habitats that didn't exist there, and which would allow other species that would normally not be able to survive there to colonize.
Right. But so are these mesophotic region fish, are their populations then linked to the shallow water fish? That is, if you overfish one of the shallow water fish that the mesophotic fish depend on as a food source.
That's a really good question, and there's a hypothesis called the mesophotic refugia hypothesis, which states that mesophotic fish that live deeper can replenish shallow water populations that have been extirpated due to overfishing or bad storm events or pollution or so forth. And so that's one of the questions that we're trying to see is this possible? Do the deeper waters actually act as a refuge for the shallow reefs? And I think the answer may be sometimes. A lot of fish are shallow water only, or they only occur down to a certain depth. They don't go all the way down into the mesophotic, and so unless you can persist below the impact area, you're not going to be able to recolonize. But there's a lot of work going into that question right now.
Excellent. And we'll talk some more about that here when we come back. We're going to take another break now. You're watching Likeable Science here on ThinkTech Hawaii. I'm your host, Ethan Allen. With me today in the studios is Joshua Copus, who's with the Hawaii Institute for Marine Biology. We're talking about his diving, research, coral reef fish, et cetera, et cetera. We'll be right back.
Aloha, Yappers. This is your host, Kingsley, for the Yap Show. Every Friday, 3 p.m. to 4 p.m. Eastern Time. You can catch us here live, ThinkTech Hawaii, and then later on we upload to our YouTube channel. We talk about youth issues, policies, youth programs, and how to transition yourself into adulthood.
But this was like a sign, I guess. Hey, life's like, hey, right. Now's your chance to go back to school, which I'm doing.
Catch us here again live, 3 p.m. to 4 p.m. Hawaii Standard Time. Aloha.
And you're back on Likeable Science. I'm your host, Ethan Allen. With me today in the studio is Joshua Copus from the University of Hawaii, the Institute for Marine Biology. We're talking about his work on coral reef fish, particularly these coral reef fish that live in the so-called mesophotic or mesophotic region at some hundreds of meters, a few hundred meters deep, not the shallow water fish. And this is apparently a largely unknown area. A lot of new species are there, species that have never been seen before. So I'm going to play a little devil's advocate here. But sort of, why should people care about this work? You know, these are fish you never see, right? No one ever really sees a lot of these fish. There's a relatively small fish. They live in these deep coral reefs. It's not like the big tuna that have a tremendous commercial importance. You know,
Well, people may care because, you know, when you're looking at the coral reef ecosystem, people really enjoy, you know, snorkeling and scuba diving on the shallow reefs. Mm-hmm. But you really have to think of the coral reef ecosystem as a whole. Mm-hmm. And these deeper reefs, we really don't know how they influence the shallower reefs. So until we really get a good, solid foundation to our understanding of mesophotic reefs, it could really be, they could be very important to the maintenance and preservation of shallow coral reefs as well.
Right, and since many of even the pelagic fish have early stages of life where they live, you know, around coral reefs, again, sort of messing with any aspect of that system could potentially impact these other species too.
Absolutely, and it's very complicated. I mean, certain life stages of fish or other organisms may live, like juveniles may live shallow and then move deeper as they get older, or vice versa. Mm-hmm. And so it's a really complex issue, but unless we really don't know much about these deep reefs. There's been very little work that has actually been done on them, but until we really understand, we don't know. And I think that's why people should care, because we need to know whether or not they're
right
and how important they are to the shallow reefs. I
was talking with a guest on an earlier show about sort of what we're doing in terms of changing our climate, and basically we're conducting sort of a vast geoengineering experiment, knowing only a handful of the variables and relationships involved, and knowing there are a lot more, but not understanding what they may be, how they may all be interacting. And this is sort of a piece of that same work, basically, is yes, we're screwing around with the coral reefs in various ways by dumping pollution onto them, by heating up the water, at least in the shallow parts of them, and in various ways altering the species composition, and what impact that's going to have downstream, not necessarily tomorrow, but the next decade, the next century, is largely unknown at this stage.
Absolutely. There's a lot of unknowns as to how resilient the coral reefs are going to be. There's a lot of, they're very uncertain. There's a lot of people who think that there's not going to be many coral reefs left in the near future.
Right.
And so the more we know, the better we'll be able to handle and make the changes that we need to make in order to manage this very valuable ecosystem. I mean, when the coral reefs go, a lot of food fish will go with them, you know?
Right.
So.
Now, in geologic terms, though, coral reefs individually are sort of transient, right? Because as this ocean goes through this rising and falling cycle, they either get exposed and eroded away or buried or become too deep. But coral reefs, because they are living systems, basically continue to exist during during these changes, right? By either building new reefs or
Right, and so a particular individual may die,
Right.
but the genetic diversity needs to persist in order to repopulate as the sea level rises again and so forth. So yeah, they do see in geological time, and we're talking 100,000 years, changes in where the coral lives. But that doesn't necessarily mean that the genetic diversity within a population will die off.
right.
And that genetic diversity is really what keeps the population healthy.
Right, absolutely. Yeah. The coral reefs are sort of as far away from a monoculture as you can get.
Yes.
They're incredibly complex systems of lots and lots and lots of different organisms of lots and lots of different types, all interacting and sort of dancing together in a really amazing.
Yeah, more so than I think we can really even envision.
Right.
In our minds, it's a really complicated system.
But there are various changes too that can happen from different sorts of events, like an uplift event that can fairly rapidly, in geologic terms, change a coral reef. We have the one spot over in Kailua where there's an uplifted coral reef there. It's quite impressive.
Yes.
Quite a few feet of coral reefs sitting up on land.
Yeah, stuff like that, or volcanoes can come through and,
Right.
The lava can wipe out a reef in a certain section, but you're talking about a small geographic area compared
Right.
to the whole picture of the reef. So if, for instance, you have uplift on one side of an island, but you have coral reef around the entire island,
Right.
then that reef can recover.
Right.
But if you've damaged and destroyed the reef largely around the entire island and compound that with uplift which is probably not a great example because we're talking human impact versus geological time, but the idea would be that the more impacts you have, the more likely that you're going to have a reef that will not persist.
Right. This is a whole issue. I mean, it goes beyond coral reefs. It goes to almost any ecosystem, and indeed into human systems that you have only, systems are only resilient to a certain extent,
Right.
and can take a certain number of insults and hits of various sorts and bounce back. But if you pound on them long enough from enough different directions, sooner or later, you get irrevocable change in them.
Absolutely, and we're seeing that all
Right.
over the world in every ecosystem, not
Right.
just coral reefs.
Right.
And yeah, there's a tipping point
Yes,
on the
and
point of no return.
Right, and it's a little perhaps frightening to think how, because again, of the unknowns here, we don't really know how close we are to hitting that point.
Right.
And at some point, or whether we may have already hit it indeed. Right?
There are probably
Right.
examples where we have
Right.
reached the tipping point, and there are probably examples where we have not. But again, we have so much more work to do before we really understand what's going on.
Right, it's amazing when you find out what would seem to be very basic information. So I was reading an intriguing piece the other day talking about the microplastic in the ocean, and they point out for years people can calculate roughly how much of this stuff should be being dumped into the ocean, and they can sample it in the water in places like the Pacific Gyre, and the numbers don't match at all. There's a huge amount that's being dumped in there that's not sitting there in the water. And of course, finally, actually somebody said, well, gee, let's look in the sediment beneath. And sure enough, the sediments all around the world, I mean, from the North Atlantic to the Mediterranean to the South Atlantic to the Indian Ocean, are apparently packed with a lot of polyester fibers, little microspheres of various sorts, all kinds of the microplastic debris. The sediments holding, in some cases, thousands of times the concentration of the water. So again, and apparently this is being done on a global scale. Whatever's living in those sediments, again, is presumably ingesting this plastic, which may not be a good thing for it.
Yeah, they'll have to either adapt or they will not survive.
Yeah. And I mean, we see the more obvious examples of it, of course, in bird populations on the islands eating so much plastic that fills their crops, and they can't fly and and
Yeah.
nutrition. So there's a lot of larger scale stuff here. So do you see your research as offering sort of pathways for hope in this, out of this gloom and doom scenario, in the sense that the better understanding hopefully will enable us to make better choices?
I think that every piece of information that we gain will give us more tools to battle the problems that we have. I hope that my research can contribute to that in some way, but until I know what the results are, I guess I can't speculate too much on that. But yeah, every bit of information that we have will help us be better stewards, I believe.
No, and it's very much the way science runs, by tiny incremental advances typically, and you get a lot of tiny incremental advances, and you realize you've actually gained some significant body of knowledge.
Yes,
even
exactly.
though each little piece doesn't seem like it really means very much by itself.
Right. Yeah. Even some of the little research projects that some people may think, why are they studying that?
Right.
They add something valuable to our overall knowledge, and eventually it will help in
Right.
some way.
I've been asked, why I spent 6 years of my life studying color vision in one particular African cichlid fish.
Exactly.
And, what did that do for the world? How did it make the world a better place? And in and of itself, probably not much, but we know a little bit more about fish color vision through that work.
Exactly.
So the other people in your lab then are doing related work. Can you maybe sort of finish up with a little bit bigger picture of where your lab is headed as a whole?
Well, our lab largely is studying phylogeography and species relationships across essentially the tropics of the world. We work in the Red Sea, the Indian Ocean, the Pacific, and to a smaller extent the Caribbean. But yeah, we're all really on that same path to try to figure out how species are related, how populations are connected, and all really working towards an understanding of, okay, if a fish disappears from one location, where are the next generation of fish going to come from? Have we lost that fish at that location, or can we get recruits from neighboring islands and so forth? So pretty much the whole lab is really focused on that type of research.
And that presumably will also inform sort of the more immediate issues of some of the commercial fish stocks, and there are many cases being heavily overfished now, and what are their prospects for bouncing back, and what strategies we might use to help them recover faster.
Absolutely. We do a lot of work with the fisheries, both here in Hawaii and other places, and so yeah, that is one of the big concerns, is what's going on with the fish that are they being overfished or are they not, and where are the next generation of fisheries fish going to come from? Yeah.
So that's your work really then does begin. We can begin to see that the tie-ins to some larger, very human-centered questions here.
Oh, absolutely.
Because clearly the world at this point really depends on a lot of seafood. I mean, it's a huge source of protein for many, many, many people around the world, and if we start messing up the coral reefs too badly and the fish stocks go down, we're all the poorer for it.
Oh, absolutely.
And lots of people are going to be in big trouble then. So, well, that's wonderful to know that that your work, which seemed when I first started talking to you very isolated, but we can see it really does tie into a lot of interesting other larger sort of global environmental ecological issues, and helps weave into that larger fabric of science so well. Well, that's very nice. I very much appreciate your being here and taking time today to come on down to the studios and share your work with us. And I look forward to maybe hearing more of your work as you progress along and seeing what it is you come up with. And I have found this to be an enriching conversation and very enjoyable.
And well, thank you for having me.
Well, thank you very much, Josh. We're about wrapped up with Likeable Science here for today. I've been here with Joshua Copus from the Hawaii Institute of Marine Biology at the University of Hawaii. We've been talking about his diving and deep sea research and the biogeography and phylogeography of coral reef fish and how that all ties into larger world problems. I hope you'll come back and join us again next week. Thank you very much.